Histology is a branch of anatomy that deals with the microscopic structure of animal and plant tissues. Histo means tissue, and ology means study, which literally translates to study of tissues. Tissues are a group of cells that have similar structure and perform specific function. The cell is the basic morphological and functional unit of all living things. Parts of the cell. Although some cells may vary in size, shape, and form, they all share a common basic structure. First, cell membrane. Cell membrane, also called plasma membrane, it is a thin membrane that surrounds every living cell. The cell membrane is composed of phospholipid bilayer. It is composed of globular polar and hydrophilic head, and two slender nonpolar hydrophobic tail. Protein molecules account for about half of the mass of the cell membrane. Proteins that span the whole thickness and project out of both surfaces of the cell membrane are the integral proteins. And proteins that are simply inserted into, or are loosely bound to, the outer or inner surface of the cell membrane are called peripheral proteins. Cholesterol molecules in cell membrane may vary in number from few to as many as phospholipid molecules, and it is found in irregular spaces between the phospholipid molecules. And lastly, glycoproteins that are found on the surface of the lipid bilayer of cell membrane. Within the cell, there are many structures that are enveloped by membrane identical to cell membrane with the same trilaminar entity, which is called the unit membrane. Many cells form specialized junctions that enable them to adhere to one another or with the extracellular matrix, or communicate with each other. Junctions that tightly bind cells to each other or to extracellular matrix are classified into occluding and adhesive. There is only one form of occluding junction, the zonula occludens or tight junction. But there are several types of adhesive junctions, like zonula adherens and desmosome or macula adherens. Junctions that enable cells to communicate with each other are exemplified by gap junctions and chemical synapses. Cytoplasm. Cytoplasm refers to the homogeneous substance that fills the space that is bounded externally by the cell membrane. The liquid found inside the cells is called cytocell or cytoplasmic matrix. Cytocell provides a suitable environment for the organelles in order to perform their function. Practically all cells have organelles. The only notable exceptions are the mature red blood cells and the lens fibers that comprise the lens of the eye. Cytoplasmic organelles include, first, mitochondria. Mitochondria is often hot-dog-shaped, but they can alter their shape and become rod-like, filamentous, and spherical. Under a lectin microscope, a mitochondrion is seen to consist of a wall that encloses a space or cavity called the intercrystal space. The intercrystal space is filled with amorphous substance called the mitochondrial matrix. The mitochondrial wall is made up of two layers of unit membrane. First, the outer membrane or outer leaflet. Second, the inner leaflet. The folds of inner leaflet forms the cristae mitochondriales. The outer and inner leaflet adhere loosely to each other, thus, forming an empty space called intermembranous space. The intermembranous space extends into the cristae's intercrystal spaces. Ribosomes. Ribosomes are seen under electron microscope as small electron-dense granules that occur singly or in small clusters called polyribosomes or polysomes. Ribosomes and polyribosomes either lie free in the cytoplasm, which are called free ribosomes, or are attached to the surfaces of endoplasmic reticulum called attached ribosomes. Endoplasmic reticulum. The endoplasmic reticulum consists of two contiguous regions. First, with the attached ribosomes and polyribosomes called the rough endoplasmic reticulum, and second, without attached ribosomes called smooth endoplasmic reticulum. Golgi complex. The Golgi complex consists of several layers of membrane-bound, smooth-surfaced, and flattened tubes or cisternae that are stacked on top of each other in a semicircular manner. Lysosomes. Lysosomes are chemical-containing pouches that move in the cytoplasmic matrix. The pouches are made up of unit membrane which contain hydrolytic enzymes. Peroxisome. Peroxisomes, like lysosomes, are membrane-bound spherical bodies that contain enzymes. But instead of hydrolases, they contain oxidases and catalases. Peroxisomes resembles lysosomes morphologically, and it's difficult to differentiate one another even under the microscope. Centrosome. The centrosome, otherwise called the microtubule organizing center, is a dense spherical area in the cytoplasm, usually located near the nucleus and many times surrounded by Golgi complex. Nucleus. The nucleus occupies the central region of the cell. It is the largest structure inside the cell. It is commonly round or spherical, but it occurs in various other shapes. The nucleus is delimited from the cytoplasmic matrix by a nuclear membrane and is filled with nuclear matrix. Nucleolus. Nucleolus is a spherical, highly basophilic structure that is usually eccentrically located in the nucleus. It is not enveloped by unit membrane and is present only in cells that are in interface.